So we're going to start with the very beginning of what's considered genetic engineering or molecular genetic techniques or um, recombinant DNA technology. These all mean the same thing. And I'm going to start with what are considered the pioneers of genetic engineering. This is a picture of Herb Boyer and Stan Cohen, and they were some of the first to discover the restriction enzymes, which naturally occur in bacteria. These are the enzymes that when a virus, a phage, inserts its chromosome into a bacterial cell as part of its infection, the bacterial cell has these enzymes that cut up that phage DNA, and that serves as one of its defense mechanisms. And so what was done in the 1960s, they isolated and purified these enzymes from different bacterial species and then showed that they could be used to cut DNA into manageable pieces. And then because multiple types of DNA could be cut with the same enzyme, they could actually be connected together in the test tube, and then you could actually combine DNA from different species and really different kingdoms. You could combine a virus chromosome with part of a human gene. You could combine a bacterial chromosome with a plant gene. And so the idea that we could create recombinant molecules in this sort of artificial way was pretty overwhelming, because we were crossing what were considered the boundaries of the kingdoms. So at that time, they didn't have any domain, but the boundaries of different kingdoms. So the bacterial groups, we could cross and recombine DNA with human. Now, at this time, there was not much of an understanding of what we call now horizontal gene transfer. They knew that bacteria could transfer DNA back and forth between different types of bacteria through conjugation and transduction and these kinds of things we talked about earlier. But now we know that there's a lot more of that horizontal gene transfer that goes on, not just between bacteria, but plants and bacteria and humans and bacteria. Animals and bacteria have horizontal gene transfer. And we think now that this goes on all the way back to the beginning of life on Earth. But at this time, they were not aware of that. And so the ability to connect DNA from across different kingdoms was believed to be a power that was equivalent to God. And so it was pretty controversial in the 1960s. But it allowed us to, let me forward the slide if I can. What it allowed us to do, you can see here, you don't have to memorize this or anything, but we name these enzymes based on what bacteria they naturally are isolated from. And then they figured out exactly what the nucleotide sequence is. This is basically along the chromosome, what the enzyme recognizes as its substrate, and then the activity of the enzyme is to just cut the DNA at that site. So it scans along the chromosome, and when it finds its recognition sequence, it cuts. And so by combining, I'll show you on the next slide, by combining DNA, let's say you cut DNA, let's say maybe this blue DNA represents just a section of a bacterial chromosome, and then you cut DNA from a human with the same enzyme. Then when you mix them together in a test tube and you add ligase, it will seal the bonds and you can connect them together. So you can create these recombinant molecules using these restriction enzymes and then connecting them together using ligase. So this was this was one of those things that just changed everything. We could manipulate DNA, we could cut it into small pieces. There were so many possibilities at this point. Around the same time, in the 1960s, they developed gel electrophoresis, a technique for being able to separate the pieces of DNA that were being cut in a test tube. So you would take, let's say, a whole bacterial chromosome, purify it from the bacteria, and then mix it in a test tube with one of these enzymes, and it would cut it, or it could be human DNA, cut it with an enzyme. And then what you could do would be to load it on into a well for an electrophoresis gel, like an agarose gel. There's different types of gels, different materials, but agarose is one that you're familiar with. And then you pull an electrical field across that gel. It will pull those fragments through the agarose material and separate them by size. And so we use the charge on DNA because DNA has a naturally occurring negative charge because of the phosphate groups. And then DNA is invisible. It doesn't have a color, colorless, I guess it might be another word. And so we have to put some kind of stain. We have to soak the gel in a stain so that we can see the fragments. And then we can take photographs. We can get information from this. And we can also take a razor blade and just cut one of the fragments if we wanted to. Let me get a pen here. If we wanted to just isolate, if we knew that this piece right here, oops, that's not a very good circle. But if we knew a certain fragment was one that we wanted, we could separate it from other fragments, cut it with a razor blade out of that agarose and pull it out and clean it up in a test tube, get the agarose away from the DNA, and we could actually have a gene in a test tube. And then we could connect that to a different piece of DNA. So there's so many techniques and things you can do with this, but gel electrophoresis was a method that was developed about the same time as the first restriction enzyme, so that we could see the DNA that we were cutting, and we could start to estimate sizes and start to figure out what we could do with these things. And one of the main things that was done with DNA that was cut with a restriction enzyme was to isolate certain pieces and then connect that fragment of DNA to what's called a vector. A vector is another piece of DNA that carries the fragment you want, the fragment of interest is what we usually call it. So let's say you have a gene, that's your fragment of DNA that you've isolated, and then you have to attach it to another molecule. And the molecule, it's called a vector because it's going to be carrying your fragment of DNA into a host cell, and the vector is something that's going to be accepted by the host cell. So if you want to introduce your DNA, let's say it's a human gene, but you want to introduce that into a bacterial cell, you need to attach your human gene to a bacterial piece of DNA. And then when you introduce that into a bacterial cell, the bacterial cell will accept it. It will be replicated in that cell, and it will not be destroyed. So when we call these a cloning vector has to have three things fundamentally. An origin of replication that's recognized by the host cell so that the cell will copy it. It has to have what we call a selectable marker, so usually an antibiotic resistance gene. And then it has to have at least one unique restriction site that matches the restriction site or the restriction enzyme that you use to cut your fragment. So that you can cut the vector. Let's say that your fragment was cut out of the human chromosome with ECAR1, then you're going to cut the vector with ECAR1. And as you'll see on the next slide, what you're going to do is mix them together in a test tube. They will naturally stick together. So your fragment here in orange, the vector DNA in gray, and then you add ligase and it will create a slightly larger version of that molecule now carrying the human gene of interest. It doesn't have to be a human gene, it could be anything. And so we do this. This was done extensively, but this is still done. And this is called gene cloning. So it's just to attach a gene to a vector and then transform it into a bacterium, for example, or you can attach your gene into a viral chromosome and then infect cells with the virus. But cloning a gene and then getting it into a host cell is the main concept here. There are different tricks we use to determine whether or not a cell or a colony of cells contains your fragment DNA or not. One is called the blue-white screen, where you insert you use a vector that has a LAC-Z gene that is cut by your restriction site, and then when you insert your fragment, your foreign DNA piece, it will interrupt the LAC-Z gene, which will create colonies on the plate that come out white. So any colony that comes out white, it tells you that the LAC-Z gene has been interrupted by your fragment. Any colony that's blue doesn't contain any foreign DNA fragment. So it's just a trick. We use these different tricks to help us sift through all of the cells that we get on these plates. If we want to express a gene, we use an expression vector. Very similar. We have an origin of replication, we have a selectable marker, we have a unique restriction site, and what they do is they put a bacterial promoter right in front of the restriction site, so that will attract the bacterial gene expression transcription factors, and so that will cause that gene to be expressed in that cell. You don't always need the cell to be expressed. Sometimes you're just allowing the cell to carry a gene and to replicate it, but sometimes you want it to be expressed. So slightly different versions of vectors are used for different purposes. Now, when we take an entire genome and we fragment it into pieces, we can actually attach all of those pieces one at a time to separate vectors and introduce each vector plus piece of DNA into host cells. So what we create when we do that, it's called a library. A library is the entire collective group of clones. Clones are bacterial cells or yeast cells typically. They could be whatever host cells you're using, but bacteria and yeast are used very commonly as host cells. And so you fragment up an entire genome, you mix all those fragments with a lot of vectors that have also been cut open, and then each fragment from your genome, let's say it's a human genome, will attach to one vector molecule. So you'll have all of these vector molecules carrying a different piece of your human genome. And then what you do is you take all of those and introduce them into host cells so that each host cell, bacteria or yeast, is carrying one vector plus one fragment of DNA. So collectively, all of the clones in the collection contain all of the fragments that make up the entire human genome. So that's called a library. And libraries have been made for every virtually every organism. And if you make a library starting with the whole human genome, or the whole genome of any organism, it's called a genomic library. And what you do with a genomic library typically is you sequence it, for example. So you can put it through one of those DNA sequencing processes. Each clone separately, one at a time, is put through sequencing. And when you have completed sequencing all of the DNA from all of the clones in your library, then you have sequenced the entire genome. So that's how genome projects, they're still done this way. The other kind of library is called a cDNA library. cDNA libraries they represent just the genes from a genome. And the way that you can get the genes out of a genome without getting all the repeat sequences and such is that you start with a tissue and you extract the mRNA from the tissue. And then you do a reverse transcription to make DNA from that mRNA. Because remember, mRNA represents the genes that are being expressed at that time in that cell tissue type. And so if you do a reverse transcription, you make DNA only from the mRNAs that are in that tissue at that moment, then what you have are DNA pieces, we call them cDNAs, that represent the genes that are being expressed in that tissue at that moment. And so cDNA libraries are made from particular tissues. So you would have a cDNA library made from brain tissue that would represent all the genes expressed in that brain tissue. You could make a cDNA library from muscle tissue, and that would represent all the genes being expressed in that muscle tissue. So those are tissue-specific libraries. A genome library would include everything, including all the junk sequences. So sometimes you want to see the whole genome as it is. Sometimes you want to sequence just the genes that are being expressed in a certain tissue type. So what you're doing when you make a library is you're breaking up, this is what this is, you're breaking up the genomic DNA here into pieces. You're attaching each piece to different, well, they're all copies of the same vector. I don't know why they showed these all different. All the vectors are the same size, so I don't care for the way they did that. But each of the pieces can be somewhat different sizes for your fragments. But they're all being attached to vectors of the same type, just many copies of the vector, and then transformed into host cells and then plated out on plates. And then we just store them in a freezer, because bacteria can be stored in a freezer and they do fine. So libraries allow us to have one type of cell, bacteria or yeast typically, carrying one piece of a genome or one gene from a tissue type. And that way we can use sequencing, we can use other techniques. Usually sequencing is kind of a good way to think about why we would make libraries. All right, so that's the end of this segment.